In this exam application tutorial, we're going to look at the topic of enzymes. The question for 6 marks is use the lock and key hypothesis to explain why amylase works best at 37 degrees Celsius. This is another fairly loaded question with lots of sub-questions hidden away. And you have to address every part of the question to make sure you're unlocking every mark available. So firstly, we must describe what the lock and key model or hypothesis is, then explain how high temperatures affect an enzyme's shape, and then explain how this affects the lock and key model. This is my interpretation of why amylase works best at 37 degrees Celsius. You see, if you start explaining why it works best at 37 degrees Celsius, it's kind of misleading. What it's really asking you is why will it not work well at temperatures above 37 degrees Celsius? So firstly, let's describe the lock and key model. First thing to say is an enzyme has an active site, and you can see that over here. So here's our enzyme with its active site, and here's our substrate, the target molecule upon which the enzyme acts. Now you'll notice that the active site has a complementary shape, in other words, like a lock and key, to a specific substrate. So that's what you need to say next. So an enzyme has an active site with a complementary shape, very important words, to a specific substrate. And it's important to say specific because enzymes have a relationship with a specific substrate. You can't just assume that any enzyme can help break down any substrate. So you can see that complementary shape in action as the substrate and enzyme move near each other and bond together. The final part of describing the lock and key model or hypothesis is saying after bonding to the active site, the substrate is broken down to form products. So that's what you can see happening here. You can see the substrate breaks down, forms products, and is released from the active site. So just remember, the lock and key model is an enzyme has an active site with a complementary shape to a specific substrate. After they bond, the substrate is broken down to form products. So as they have mentioned amylase in the question, I think it's wise to actually deal with amylase specifically and say, for example, amylase would break down starch into glucose. It's just letting the examiner know that I'm reading the question and answering every part of the question. It shows you have flexibility in your answering. So now let's link the idea of temperature change to an enzyme's shape and activity. You see, 37 degrees Celsius is the optimum temperature for amylase. They talked about 37 degrees Celsius here, so I must include that in my answer. 37 degrees Celsius is the optimum temperature for amylase. That means it's the temperature at which amylase works best. If temperatures get high, or high temperatures, cause amylase to denature. In other words, when we increase the temperature, the bonds holding that polypeptide chain, which is folded into a three-dimensional shape, start to vibrate and eventually break open, and the protein's shape changes. More importantly, the active site changes. This is called denaturing. Most questions on enzymes require you to use the word denature, so when you're at a loss, just write denature down in your answer. But be aware that enzymes only denature if they're too hot, not if they're too cold. Whereas enzymes can denature if pH rises or drops, but temperature only if it rises. So now that the enzyme is denatured, starch will no longer fit into the active site. So now I've addressed every part of this question. I've explained the lock and key hypothesis. I've explained amylase and what it does. I've said that it works best at 37 degrees because it's its optimum temperature, and I've explained what happens if you go above this temperature. And of course, I've linked it to the lock and key hypothesis because I'm saying above this temperature, that substrate will no longer fit into the active site, so our key no longer fits into the lock.